My name is Sandy Chan, and I'm a process engineer at the drinking water company Sydvatten, located in the south of Sweden. I will talk about how we follow bacteria in different drinking water scenarios, and also talk about the question if we can use flow cytometry for process control. First, shortly about Sydvatten AB, which provide water for almost 1 million Swedes. We have 3 different lakes. Lake Bolmen, located here, Lake Vomsjön, located here, and which is our main sources. And then we have Lake Ringsjön, which is our backup. In our system, we have also 2 waterworks. One that is a traditional chemically treated surface waterwork located here, and also one that is an artificial groundwater infiltration work. We are a drinking water producer and have mostly responsible of the main water pipes of the cities, as you can see, these cities in Sweden. And we don't have the smaller and more fine part of the distribution system. Other companies or municipalities are responsible of that, but still it contains our water. So I have been working as a process engineer at Sydvatten since beginning of 2019, so not for so long, I would like to say. And my main focus is basically the living things in our water, where I have some as one of my tasks to apply research into our daily work. So I would like to tell you a story about how Sydvatten started using flow cytometry, which is also my personal story into the world of drinking water. But it's also the story how flow cytometry for drinking water got introduced in Sweden. So before working as a process engineer, I was an industrial PhD student at Sydvatten and the research company Sweden Water Research. My main research focus was on biofilms in our drinking water system, especially about how slow sound filter worked in the waterworks. So in my research, I apply flow cytometry to answer some of my questions. And I used a more advanced data analysis of the flow cytometry data to describe the results, combining both the total cell count and the fingerprint, meaning the HNA and the LNA. So in the dot plot, as you can see here, each dot in this square represents a bacteria that was measured from this sample from the flow cytometer. And by comparing each dot location with another sample's dots, other sample's bacteria, we can find similarities and differences between these samples, and not only look at the total amount of cells, but also we get a snapshot of the bacteria in these samples and what are similar or not according to how much dye each bacteria has, which is an indication of how much DNA they have. So a simple way to put it is that it's basically that the game to find differences between 2 pictures, and everything by combining water samples in this case, and everything can summarize into a plot. For example, an NMDS plot that I will show you. So in an NMDS plot, every sample which is represented as a dot is the dot plot of the flow cytometer. Is presented here. So if 2 dots are close to each other, it means that the results from the flow cytometer is similar. If the dots are far apart, the total count and also the fingerprint from flow cytometry is not so similar. So I used this method to study 3 different slow sound filters at the waterworks based on maturation. One was a well-established slow sound filter, a full scale, and 2 new filters, also full scale, where we have one that contains old washed slow sound filter sand on the top with new sand at the bottom, and a totally new slow sound filter containing only new sand. So looking at the data, we see that this is the influent, the water going into these 3 different flow sand filters, and they are clustering together as one group, which is not so surprising, since this filter receives the same water from the same treatment plant. The interesting part is the effluent water. Here we have the effluent water sample leaving the established filter in green over time, and we see that they are very similar to each other since the dots are very close to each other, and maybe most importantly is different from the influent water. So the biofilm living in this filter is able to transform for the water from here to here based on flow cytometry. The new slow sand filter that was constructed with both the old and new sand ended up here in this plot. And as you can see, this cluster is more spread over time and different from the established filter. And the last filter is a sand filter with totally new sand that actually forms 2 clusters over time, with the end of the period forming a cluster quite close to the influent water. So here we see that flow cytometry was able to distinguish water produced by different slow sand filter. And an interesting twist into this whole thing was that these clusters here was also related to E. coli content. So we see that using flow cytometry, we can have a great potential to reach our goal, which is always maintaining a good drinking water quality from waterworks to consumers. And we would like to do that in a rapid way, and it should be simple. Thereby, we can save money, but also, most importantly, we can save water. FCM can also help us to get a better understanding of microbiology in our drinking water system, and can be a backbone for decision making and troubleshooting. So we see that BactoSense can have all these elements that we are looking for. So let me show you our summer plan with our 2 BactoSense that we have bought. So we are planning to use BactoSense to look into different events or different parts in our system right now. So for example, right now we have the BactoSense measuring the incoming and the outgoing water of a reservoir. And we have been doing so during the autumn and during the winter, and we would like to see how it will change during the warmer month here, where we know that we can have some trouble with heterotrophs in the system. So we would like to see if we can use BactoSense to see when we need to change the water level a little bit more drastic in this reservoir to avoid microbial growth in a smart way. We're also planning to see the effect of flushing and change in flow direction in the pipe system, and see if BactoSense can see that. So now I would like to show you an example where we have put these 2 BactoSense in the field and looked into a pipe leakage. So last summer, we have a pipe leakage in the middle of a field, as you can see here in the picture. And as always, after changing the broken section, we need to flush until the affected pipe section is clean, meaning no coliforms, no E. coli, and also low heterotrophic plate counts. So the question is here is that can we use BactoSense to see when we have achieved a good quality, meaning that we have flushed enough. So we had a section of a pipe around 10 kilometers with a diameter of 800 millimeter that got a leakage, approximately here. So we put 1 BactoSense upstream of the leakage, not affected by the leakage, and we put another BactoSense downstream of the leakage. So the main goal was to generate data and see if we can see when we have flushed enough after we have fixed the pipe section. But we also wanted to test out the BactoSense a little bit more, how portable are BactoSense and how easy are the instruments to install, and is it suitable for all field personnel. So here you see the results for the 2 BactoSense. On the y-axis, you have the total cell content per milliliter, and on the x-axis, you have the measuring point. I apologize for the messy x-axis, but I just want to point out how often we were measuring, which was every hour. And that should be compared to our grab sample, which we also did, which was only once per day. So the blue line is the measurement from the sample, meaning that the BactoSense sitting downstream of the affected area. And the orange line is the reference point, the point where it was not affected by the leakage. So as you can see, we have a very high and not stable system the first day, according to the total cell count. But the total cell count stabilized quite quickly after around 1 day. But looking into the data a bit closer, using a similar approach that I talked about before, with the slow SAM filter, we see that dot plots that we can see changes in the dot plot within the first 130 hours, so around within the first 5 days. We have again a similar plot where we have the reference sample here in a closed cluster in orange, meaning no change of the water sample, which was expected. The color change in these dots are reflecting the time. So black is the beginning of the flushing and white is at the end. So in total, we flush for a little bit more than 2 weeks. Looking at the sample point, we see that we have a change in the first 6 hours, quite drastic change, but does not form a closed cluster until 5 to 6 days. And I need to say that these results are normalized results based on total cells, meaning that we have normalized the total cell content to be the same in each sample, in each dot plot. So the changes we see here is how the bacteria is moving in the dot plot and not in number of counts. So if you are very interested in your data, which I am, and you would like to dig into detail, you can also get to know where in your dot plot you have the biggest changes. So in my case, in this leakage test, we have the largest changes in the dot plot in this area and also in this area. So by having this more advanced data analysis, you can also learn how to look at your data when you are looking at your BactoSense live. I want to acknowledge my great collaborator, Dr. Nicholas Gadot, and also my former PhD supervisor, which I have many collaboration with right now, Dr. Catherine Paul, for these exciting flow cytometry data projects. And this flushing event is a part of Mons Samour's master thesis that he will present during this summer. But going back to our question for this test, we think this kind of setup with BactoSense has a great potential to follow our flushing work, for example, in a system. The results combined with a smart data analysis can be the future way for us. Our next step is to actually apply artificial intelligence on this data set and see if we can predict if there will be any changes in the data or not using the reference sample. For the more practical part, it was easy to install and move the BactoSense to the different locations that can be a little bit tricky to go to. And our field personnel didn't have any problems to check it up during sampling or changing the sampling tab to install the instrument. That was quite easy according to them. So to summarize my presentation and give you my take-home message, I would like to point out that our flow cytometry data includes a lot of information, not just the count, which is perfectly described in this saying, a picture is worth 1,000 words. From my example I show you, it is not only the total bacteria count that can be of interest, but a lot more information lays under every dot plot that flow cytometry generates. And this becomes even more important and even more powerful when we now have the ability to measure it online, which generates a more complete picture of what is actually happening. So with my take-home message, I would like to thank you for your attention. And this is also my contact info if you have any question or any suggestion after this webinar. I will be happy to receive that.